Bio, welcome to part 2 of a 2-part series on transcription and translation. So today we're going to be focusing on translation, taking an RNA message and creating a protein. Okay, so in order to do this, we need to break the DNA code. Now, I've been talking in class, kind of saying this throughout the whole year, that DNA is a code. And some of you still might not quite understand how that works, but hopefully by the end of class today, you will. Okay, let's imagine that we have a DNA sequence. And that DNA sequence is equal to TAC. And I'm going to move this down slightly. Okay, so 3 DNA nucleotides can be used to construct 3 RNA nucleotides. So you all know that RNA is, RNA is complementary to DNA. So if we have a DNA sequence of TAC, then the complementary RNA sequence is going to be AUG. Okay, 3 RNA nucleotides is what we call a triplet, or a codon. 1 codon is equal to 1 amino acid. So, our goal is to figure out what is the amino acid that is encoded in AUG. And we can use this code breaker in order to figure it out. So, what we do is we start over here, all the way on the left side, in the first letter. We know our first RNA letter is A, so we're going to go down to the A row and highlight it here. Our second letter is U, so up here we have to find the second letter and identify U here. And then our third letter is G. So within the intersecting area between U and A here, we need to highlight this G. And then we triangulate the spot where all 3 of them intersect, and we find it is right here, AUG. And this codes for the amino acid methionine, or MET. So I'm going to write that here. So the amino acid that AUG codes for is methionine. It's in red, it's special, it's called the start codon. More on that later. Okay, so we just deciphered the code. We took a DNA sequence, we translated it into RNA, and then we found the amino acid sequence, or the amino acid that correlated with that sequence. Now you might notice that there are a total of 64 codons, 64 unique sequences of mRNA triplets. And there are only 20 amino acids. So the genetic code has something that's called redundancy without ambiguity. What do I mean by that? So notice here that GUU codes for valine, and GUC also codes for valine. So valine has some redundant codons. All 4 of these codons actually all code for valine. So we can use any one of them to call up the amino acid valine. However, GUU is found nowhere else in this table. So GUU never codes for leucine or for proline or for histidine. or for glycine, it only codes for valine. So the genetic code has redundancy. There are more than one codon for every amino acid, except for methionine. Notice there's only one. But it's unambiguous. GUU will always code for valine, no matter what. Okay, so knowing this, or having this piece of information, we're going to slide over here and talk a little bit about what's happening in this image. So the completed mRNA has just left the nucleus, and in order to understand what's happening to it, we've got to label some components. Oh, I already labeled this. So here we have a ribosome. A ribosome is composed of two subunits, the large and the small, that come together and clamp down on an mRNA message, and then they start to slide across it in this direction. So you can either think of the ribosome moving this way or the mRNA message moving this way. They're both kind of the same thing. All motion is relative motion. Okay, so our ribosome is going to come together, the large and the small subunits. They're going to clamp down on this mRNA message, and they're going to start to move along it until they come across a codon that's called the start codon. And then they are going to initiate translation. So as soon as they run into the first AUG, which codes for methionine, so this amino acid at the end is going to be a met. We're going to start to read the message in groups of 3. Okay, but before we really get into that, let's talk about what these little green diddlies are. So this is what's called a T. Actually, I'm going to make it lowercase. Why is my eraser so giant? This is what's called a tRNA, where the T stands for transfer. Every tRNA is bound to a specific amino acid. And every tRNA is going to have a group of 3 nucleotides called an anticodon. That will make sense in just a minute. Okay, so know that there are, just like mRNA codons, there are 64 tRNA anticodons. And just like mRNA, tRNA is going to have redundancy without ambiguity. So whatever this triplet is, it's going to be specific to the amino acid that's bound to the top. Okay, back to the message. So notice what's happening here. We have 3 sites in our ribosome. Let's label them. The A site is called the arrival site. The P site is the polymerization. And the E site is the exit. So we are catching our ribosome here in the middle of translation. It has started creating an amino acid sequence. So these amino acids are linked together by covalent bonds called peptide bonds. And because this is not yet folded, it's just a polypeptide. And how is it being synthesized? Well, notice that there is an empty tRNA right here. This tRNA used to be bound to this sequence right here, but it passed off the sequence to the current tRNA that's in the polymerization site, and it is currently leaving the exit site. But notice something interesting about the tRNA. It has an anticodon that is the perfect complementary match for the mRNA codon. Okay, so currently in the polymerization site is a tRNA with the sequence AAA. It is complementary to the sequence UUU. We can go down to our chart here, our nifty chart, and say UUU, oh, phenylalanine. So we know that this amino acid, currently right here, must be phenylalanine, PHE. Okay, so currently this tRNA is carrying the polypeptide chain. But in a moment, this tRNA anticodon is going to bind to this mRNA codon through complementary base pairing. This is a sequence AUG, which is AUG, so this must be methionine. So this tRNA will bind to, the anticodon will bind to the codon through complementary base pairing. And then what's going to happen is from the polymerization site, there will be a peptide bond formed between this phenylalanine and this methionine, and this bond will be broken. So now the ribosome will slide over one position. This amino acid will now be in the E site, and it will be ready, not amino acid, sorry, this tRNA will be in the E site, and it will be ready to jet away. This amino acid, why do I keep calling them amino acids? This tRNA will be now in the P site, and it will be carrying the polypeptide chain attached to methionine, attached to it. And the A site will be open and empty, and there'll be another sequence of 3 RNA nucleotides here ready to bind the next transfer RNA anticodon. And so on and so forth. This chain will continue to grow until the codon is reached that is one of these 3 stop codons, UAA, UAG, or UGA. This will cause the ribosome to fall off the mRNA message and release the polypeptide chain, which will then, if this is in the cytoplasm, be folded into a functional protein. So that's it. That's the secret of life. Now, the truly amazing thing is here that this genetic code is universal. So the sequence UUU codon, RNA codon, in any living organism always codes for phenylalanine. The sequence UCU always codes for serine. In any living thing, which is just amazing. Like bacteria are using the exact same sequence of RNA nucleotides to construct proteins as you are. So you can take a gene from any organism, any sequence of nucleotides, and stick it into another organism, and they will start to make RNAs, and then they will start to make those proteins. So you can take the sequence for green fluorescent protein in a jellyfish and put it in a mouse, and then a mouse will start glowing fluorescent green, and that's been done. You should look it up on the internet. So that is the secret of life. Ta-da!